Johar and welcome to this presentation on the topic cultivation of wild edible termitomyces growing in the forest of Jharkhand. Abstract. Fresh mushrooms and cinemata of termitomyces were collected from the field during rainy season. Dikaryotic hyphae from these parts were isolated aseptically and were inoculated in the different culture media prepared, as well as on the boiled sterilized wheat grains taken in the saline bottle for obtaining pure culture. There was mycelia growth on MA medium, and this culture was used for spawn preparation. Spawning was done on the specially prepared compost manure as well as on the mud taken from the termite cell and on the paddy store. There was fruiting body development on the culture medium. Keywords: cinemata, wild edible termitomyces, cultivation, compost manure. Introduction. Wild edible mushrooms are one of the important natural resources on which the local people of all nationalities rely heavily, and these mushrooms certainly play a role in improving the food nutrition. Termitomyces species is an important source of enzymes of industrial importance, such as glucanase, amylase, and cellulase. Antioxidant compounds such as polyphenol and vitamin C. Culture studies of the genus Dermatomyces hemi have been conducted on mycelium from Com sporodokia. Isolation into pure culture of Dermatomyces species was done from the asexual fruit bodies growing on the fungus garden. Dermatomyces was isolated in pure culture aseptically, placing nodules on malt yeast stacked agar petri dish containing MYA. Dermatomyces hemi pure culture were isolated from the asexual fruit bodies growing on and near the termite swarm by tissue culture techniques. In three different media, Ghost et al., Modest, and modified mold stack agar. Termitomyces species of mushrooms are the most liked and consumed by the local people of Jharkhand. Villagers collect a lot of wild edible mushrooms for sale or consumption during rainy season. The culture methods of these edible mushrooms have not been developed, and there is rapid deforestation due to various factors. There is a possibility that these wild edible mushrooms may be lost forever in near future from the forest if they are not preserved immediately. There are four main species of Termitomyces growing in Jharkhand: Termitomyces clypeatus, Termitomyces albuminosa, Termitomyces hemi, and Termitomyces microcarpus. Termitomyces clypeatus. The distinguishing character of this mushroom is the prominent cone-like pointed dark projection at the center of the cap. This mushroom is locally known as chico. Termitomyces albuminosa. It is locally known as Bada Kukri, which means big mushroom. It has a patch of veil present at the top of the pilus, and the skin of the cap peels off. It is found in the first and second week of September. It takes almost 24 hours to develop into a full matured fruiting body as it is shown in the slide. Dermatomyces hemi. In Jharkhand, this mushroom is known as patiari, and it is found most abundantly. It comes up exclusively from termites mound. Dermatomyces microcarpus. Among the four, this is the smallest in size, but very tasty mushroom. Material and method. Collection of fresh mushrooms. Fresh fruiting bodies of Termitomyces hemi and Termitomyces albuminosa were collected from their natural habitats during rainy season. Their cinemata was also dug out from termites' mound. Following culture media were prepared and autoclaved. PDA medium, Gauss et al. Medium, modified malt strike agar medium, Hesium modest medium, mycelia culture on the media. After autoclaving media were prepared, poured into test tubes and slants were made. In these slants, mycelia was transferred aseptically in the laminar airflow chamber. Mycelia was also directly transferred on the boiled sterilized wheat grain taken in the saline bottles. Taking out the dikaryotic mycelium. For both the mushrooms, dikaryotic hyphae from calf and cinemata were used to obtain pure culture. Boil sterilized wheat grains. Mycelia growth. Only Termitomyces albuminosa showed mycelia growth on modified malt strike agar medium. We can see the mycelia growth on MEA medium. There was mycelia growth of Termitomyces albuminosa on the boiled sterilized wheat grain also. We can see the slide. Preparation of substratum. The study of natural habitat of these wild edible mushrooms revealed that these mushrooms grow in the soil enriched by humus of decaying leaves. Therefore, to grow these mushrooms, compost manure was prepared. In the compost, leaves of Shoria robusta, Syzygium cumini, Mangifera indica, Basia latifolia, and Chelicera oleosa were added as they are the main trees in the forest of Jharkhand. Spawning. Spawning was done on the compost manure prepared, on paddy straw, on mud taken from termites' mound. Mycelia did not develop fruiting bodies on the compost. There was fruiting body developed on the culture media. Discussion. The wild edible mushrooms vary in their temperature, humidity, and nutrient requirement and habitat, and occur only in particular month of the year. For example, Dermatomyces albuminosa occur only in particular month of the year. It occurs in the first week of September. Some mushrooms grow only in the particular habitat. Similar report has been given by Steven, as he has stated variation in timing, sequence, mechanism, and regulation of the basic steps yield the incredibly varied life cycle encountered within the fungi. Kaun has reported mushrooms can utilize enormous variety of organic substances as energy source. For the growth and reproduction of mushrooms, nutrients should contain carbon, nitrogen, sulfur, phosphorus, and trace elements. Some require an extraneous supply of vitamins. They thrive well in a warm and humid environment. The temperature range for their growth is very wide. The state of the substrate also affects their growth and reproduction. pH range also affects growth. Oxygen and moisture are other factors affecting growth and reproduction of mushrooms. Majority of mushrooms found in Jharkhand are symbiotic. Some mushrooms require special nutrient for their growth. Through the experiment, it was found that not all mushrooms show mycelia growth on the culture media. Summary and conclusion. Results showed that Mycelia of Dermatomyces albuminosa grows on modified mole stack agar medium and on boiled sterilized wheat grains. Therefore, domestication of these mushrooms is not an easy job. More research work is needed to cultivate them in an artificial condition. These are my acknowledgements. These are the references. Thank you.